is titled Chemical Multiverse and Diversity of Food Chemicals. My name is Juan Felipe. This work consists on the application of cheminformatics techniques to food science, expanding the emerging field of food informatics, and are the preliminary results of my master project. It was developed under the direction of Dr. José Luis Medina Franco at DIFACQUIM in the Universidad Nacional Autónoma de México. As an approach for food chemical multiverse, we used the largest public database of food chemicals called FoodDB. It's maintained by some Canadian institutes and contains more than 70,000 compounds from almost 800 different foods and information such as physicochemical, biological, and organoleptic properties. The state of the art is such that there is a previous report of the characterization of food chemical space by a research group 5 years ago, when the database only contained almost 30,000 compounds, and the chemical space covering and the physicochemical properties related to oral biodisponibility of the compounds showed a broader distribution for food chemicals comparing to reference datasets. The molecular complexity by carbon with hybridization sp3 and the scaffold content showed also a trend to acyclic compounds. The principal objective of the current work was to characterize the chemical multiverse of food chemicals. And to achieve this, we compared the overlapping of the dataset versus FDA-approved drugs and the Universal Natural Product database in its subset A. We also compared them in terms of the molecular descriptors, the scaffold content and diversity, the natural product likeness score, the structural diversity by Tanimoto's coefficient, the chemical multiverse coverage, and the chemical profiling using the neural network approach named natural product classifier. Some of the main results are the interesting compound overlapping among the three datasets. For example, the presence of nutrients broadly used in clinical hospitalization, such as adenosine or nicotinamide, and others used in the treatment of specific diseases like morphine and ethyl and methyl salicylate, which are precursors for salicylate, acetyl salicylic acid. Sorry. Also, in terms of the molecular descriptors, we found that some patterns of physicochemical properties evidenced a trend to larger and less polar compounds, for example, in high molecular weight and high octanol water coefficient of partition. Also, a larger fraction of sp3 hybridized carbon atoms and rotatable bonds makes us think, for example, in alkylic compounds. And there is a curious pattern of 6 atoms of oxygen and 6 hydrogen bonding acceptors that could make us think, for example, in triacylglycerides. In terms of the molecular similarity for the characterization of the diversity of the datasets, we found that with different calculated fingerprints or representations, there is a higher values of similarity for food chemicals versus the references datasets that you can see in this table, and also in the cumulative similarity curves, supporting the enrichment of the dataset with fatty acids or triacylglycerides. Some other conclusive ideas are that there is a wider coverage of the chemical space or chemical multiverse for food chemicals. The most represented biosynthetic pathway predicted with natural product classifier was effectively fatty acids. And there is a necessity of further studies for the development of machine learning models and the discovery of bioactive compounds based on food chemicals. That's why we think it will be interesting to explore the data set in terms of the ring chemical space, the scaffold, the content of unique scaffolds, the content of fragments, the maximum common substructures, and to generate a public database for the novel science, for example. Also to explore computationally the biological space of potential interactions of food chemicals comparing them with FDA-approved drugs and PDB bind to develop also predictive models. Also, this project is under review in the journal SAR and QSAR in Environmental Research in their special issue for this event. And I'd like to thank CONACYT, DGAPA for supporting this project, and the DIFACQUIM research group. And there is some contact information. Thank you.
Thank you for interesting talk. We have one question from Dmitry Shulga. Is it possible to develop a score which discerns potential food chemicals among general chemical universe based on your research?
We think it's this work is a first step going to this approach. For example, knowing the natural product likeness score, would like to go in this direction.
Okay, thank you. Another question from Vladimir Paroykov. If you try to estimate toxicity of these chemicals in silico?
Okay, this is also an approach we could follow, for example, by pairing the dataset with PDB bind. This is one of the main ideas of the perspectives of the work. Also, we think that food chemicals are generally accepted as safe for human consumption. Then it will be a good source of bioactive compounds.
Okay, thank you. Another question from Henish Senderovich. In this case, does size matter? Would you have reached the same conclusions had you analyzed the food DB content which was available 5 years ago?
Of course, we based our conclusions in the previous report of FoodDB cheminformatic analysis. And we could say that there is an enrichment of some particular molecules in the previous version, from the previous version to current one of FoodDB. Specifically regarding fatty acids and triacylglycerides specifically.
Okay, thank you. We have another question from Roman Yefremov. What's about synthetic food predicted by de novo?
I think this is one of the main approaches for the perspectives to characterize the content of unique scaffolds and fragments of FoodDB, because as you know, the novel libraries currently available are based specifically on drug type compounds, and there is a novelty for natural products, but in the case of food chemicals, there are a lack of this kind of libraries. So that's exactly why we think it will be a good perspective for this work.